All righty, we are on to the second section in this class. Today's Thursday, September 16th. And we basically have three main and obviously related topics for this section. We are going to do the lymphatic system. We are then going to discuss the immune response. And then from there, we are going to move on to the endocrine system. So we are going to have a lot of material to cover in a relatively fast amount of time. I'll give a couple weeks plus to do this. However, I say this, there are two big challenges that I think people have in this. Unlike the last section where you can hold a heart in your hand, you can clearly see the blood vessels and stuff like that. A lot of the things we're going to be talking about this are going to be cellular and chemical interactions, and those are not things you can hold in your hand. So they're a little less intuitive. There's also going to be a lot more histology than there was on the last exam as well. So that's something you're going to need to watch out for as well. The other trap that students often fall into in this section has to do with the other major thing we're going to be doing, and that's going to be the group presentations. Today, we will talk about what that assignment is going to be. You're going to be randomly formed into groups, and you're going to be picking your topics for that. And one of the traps that I've seen students fall into in the past is to focus way too much of their time onto the group presentations. The group presentations are important. They're in some ways fun, and it is 50 points that it is going to be worth. However, I will remind you that the lecture exam is worth 100 points. The lab exam for this one usually is somewhere between 60 and 70 points on this. So while certainly the group presentations, Luna just got in, she snuck by you. While 50 points is important, don't spend so much time focused on that you're not preparing for the lab and the lecture exam, because those things are definitely worth more. Because you're doing group presentations, as you'll see, there is not nearly as much homework in this section as in the last one. You only have one unit review you're going to be turning in. You have one Physio-X exercise, and it's really just the one activity, activity 3 from that, and one Lapster. Because I expect you to be spending most of your time preparing for this exam and also working together on your group presentations. Also, there are few advantages to us being online, but one of them is that you're going to be responsible for, and we'll talk about this more when we get to it, you are going to be responsible for doing an outline on your group presentation that will be available for everybody to look at and study from, because this is going to be testable material. And while I don't normally like having assignments due on days when we're not in class, because it's too easy to forget about those responsibilities, I do want to give you a little extra time to work on those outlines. So we're going to have those outlines due on Friday, October 1st by 6 p.m. Make sure, again, it's just one per group. So if there's 3 people in your group, you just still need to just turn in one outline. But make sure all 3 of your names are on it, make sure your topic is on there, and make sure your references are on there. And we'll talk about that more when we get to the instructions for today. It's vitally important that it be turned in on time on Friday by 6 p.m. You can certainly turn it in early once you're done with it, because like I said, this is going to be testable material that people are going to be responsible for, and I want them to have access to it and even be able to print it out if they want to ahead of time. Because on Tuesday, when we come back after the weekend, you are going to have your group presentations. So we'll spend hopefully the whole day. If we fall a little behind on the lecture, we may have to lecture a little bit, but I'm going to try really hard that that doesn't happen. And then on Thursday of that week, you'll have your lab and lecture exams. So even though the schedule doesn't look quite as busy, there's not as much on it, it will keep you busy for the next few weeks. All right, any questions on any of that game plan wise? Like I said, I won't answer any questions on the group presentation because we'll do that all at once after today's lecture. But any questions on anything else?
So you said it's going to be 3 people in each group?
2 to 3. So again, depending on who is here and who isn't here, when we divide the groups up, usually 2 to 3 people. So that's how many is usually in a group, yes.
And then for the presentation topic, is it going to be only the ones that we studied?
As I said, we will talk about the group presentations and your topic that your group will select and all of that after the lecture today. But I don't want to answer those questions now because many of it will be answered when we go over this together a little bit later. So like I said, if you have any questions on anything else other than the group presentations, I will happily answer those. But let's save the group presentation discussion till the time when we actually talk about it.
Do we not have class next Thursday?
No, you have class Thursday. Why?
There's nothing due that day.
True. Oh, okay. You are correct. Yes. Okay. The schedule that you see here isn't the schedule of when we're meeting. You're right. There, we don't have anything due on that day. So you're right. We don't have anything due on that day, but we are still meeting on Thursday. So there is still class. There's just nothing due on that day. And what is that? Thursday, 9/23. Yeah, we still have class on that day. There's just no assignment due on that day. So yeah. Notice there's nothing due on, well, I guess your group presentations are due on the 5th, but homework wise. So, yep. So thank you for asking that. Yeah, I apologize for confusion. I just had the assignments and when they were due here. Excellent. Oh, and speaking of the exam, I hope to have it back by Tuesday. Like I said, as it turned out, both your exam and my 430 classes exam were this week. Theirs was on Monday. So I spent most of my time grading their stuff. So I haven't even really barely scratched the surface of your guys' stuff. It usually takes me about a week to get the stuff back. Once I get things graded and then curved, I will post the scores. So as soon as that is done, you will have access to it. After we've talked about it in class, then I will release the exams themselves so you can actually look at them, see what you got right, see what you got wrong. And then if you have any questions, we can discuss it at that point as well. And I'm hoping to do that by Tuesday. All right. Any other questions? All right, excellent. Then let us dive into the new material. I love your textbook in its introduction to the lymphatic system. I think they finally removed it, but for the last couple of versions, I'll be honest, I forgot to look in this newest version of your textbook. One of the things that they say at the introduction to the lymphatic system when they're talking about the organ systems is that they can't all be rock stars. And the lymphatic system does kind of get the redheaded stepchild kind of treatment when it comes to the organ systems as not being necessarily as important as some of the other ones, although certainly it does do some very important and valuable stuff for us. Of course, the primary structure of the lymphatic system is the lymphatic vessels. And what is the job of those lymphatic vessels?
They hold fluid?
True, but is their job really just to hold the fluid?
They kind of return the fluid back to the heart.
Exactly. Remember, one of the things we learned in the last section is that our capillaries are leaky. Remember, there's more force pushing fluid out of the capillaries than there's force bringing it back in. So every time exchange takes place at a capillary, a little bit of fluid doesn't make it back into the blood system. And so it's the lymphatic system's job to grab that fluid and drain that fluid out of the parts of your body and deposit it back into the cardiovascular system. And does it do it just blindly, just grabbing the fluid and pouring it back into the heart? No, exactly. Its job is to also monitor, looking at that fluid, looking for harmful things, for abnormal cells, for pathogens, foreign, abnormal things along those lines. And so those lymphatic vessels have lymph nodes that help to assist in that process. And then there are also secondary lymphatic structures, secondary because they don't contain lymph in them, but are equally important in their function, things like the thymus and the spleen, red bone marrow, tonsils, and MALT, which is mucosa-associated lymphoid tissues. And we'll talk a little bit more about those later. And as we saw in the pretty picture on the introductory slide, and as we see for this pretty picture here in your textbook, we see all of those structures. And again, they are going to contain that fluid that we call lymph. We call it lymph based solely on its location. As we talked about, that fluid and stuff, when it was in the capillaries, we called plasma. When it leaks out of the capillary into the tissues, we call it interstitial fluid. And when that interstitial fluid moves into the lymphatic vessel, then we call it lymph. So if you think about it, plasma, interstitial fluid, and lymph are all really the same stuff. The only difference, well, one major difference is the location. So obviously, something that is in the blood can affect the interstitial fluid and can also affect the lymph as well. As we talked about, this is important for two reasons. If our capillaries were continuously leaky and just leaked out forever, eventually we would lose all our blood plasma, and we know that wouldn't be good. But the other side of it as well is that it helps to reduce the amount of fluid in the interstitial tissues. This, of course, reduces the hydrostatic pressure, helping to maintain capillary exchange, and stops edema, stops the swelling of the tissues that, as we know, can cause stiffness, pain, discomfort, congestion, and issues along those lines as well. These lymphatic capillaries are large vessels that can be used for transportation, not just of fluid, but of other components as well. As we'll learn, the digestive system takes our dietary fats and puts them into lymphatic capillaries. Many lipid-soluble vitamins and things along those lines will also be carried from our digestive system back into the blood and can actually be processed by the lymphatic system. And remember, as we hinted at in the last class, if you're healthy, only about 2% of your white blood cells are actually in circulation. So that means 98% of your leukocytes are located someplace else, like in the skin, like in the liver, and many of them are housed in our lymphatic tissues, like our tonsils, like our lymph nodes, like structures like that, spleen. These leukocytes are responsible for what we call our immune response. Again, remember, we do not have an immune system. We have an immune response. We have a cellular and chemical interactions that provide protection for our body. As I mentioned, lymph is that excess fluid, that interstitial fluid, when it is located in our lymphatic vessels. The lymphatic vessels are both similar and different to the blood vessels. Blood vessels obviously carry blood in two directions. In fact, that's how we define them. Arteries move blood away from the heart, and veins move blood back to the heart. But lymphatic vessels only move blood in one direction. And what would that one direction be? Away from the heart or towards the heart?
Towards the heart.
Towards the heart, absolutely, right? Notice we talked about how veins have difficulty doing that because they have to fight gravity. They're dealing with low pressure gradients, but at least at the far end of the veins, we have the heart helping to move that. But the same is not true for our lymphatic vessels. There is no pump. There is no heart moving those things back. So it makes it more challenging. Luckily, our lymphatic vessels can use many of the same tools that our veins do. They are going to have a large lumen that can help them. They are going to have those one-way valves. In fact, typically they have more than those that are seen in veins. So they have a massive number of valves. They can also take advantage of their skeletal muscle pump. Remember, as we contract our muscles and they change shape, they squeeze the vessels and help to move. As we breathe, those changes of volume and changes of pressure in our thoracic and abdominal pelvic cavity can help to move the lymph back. These are all things that our veins use as well, but they also have another trick that our veins don't. Large lymphatic vessels can actually use peristalsis. Can someone remind me again what peristalsis is?
It's relaxation, contraction, like a wave-like manner.
Exactly. It is the rhythmic contraction and relaxation of a hollow tubed vessel to move substances from one location to another. So it is special muscle contractions for propulsion to move things from one location to another. Notice again, that's not something that our veins do. Our veins have smooth muscle, but they don't make those same kind of rhythmic contractions to move something from one location to another. But our lymphatic vessels, or at least the large lymphatic vessels, can. Now, even with all this help, it's not an incredibly efficient process. If you think of how much blood is moved through your body in a single day, and then we compare that to lymphatic vessels only move, only move about 3 liters of lymph from the extremities of your body back into your cardiovascular system a day. But just out of curiosity, how much fluid do you think you lose out of your capillaries during the course of a day?
Is it 3 liters?
Yeah, it's 3 liters, exactly. So while it may not seem like a lot, it is definitely enough. And it maintains homeostasis, which is, of course, our goal. So it doesn't sound like it's very efficient, but it is efficient enough to maintain that homeostasis and to help us to do the jobs we need to do. Now, lymphatic vessels are come in different types, similar to blood vessels coming in different types. Some of those major differences are differences in size, but also differences in location and function. Probably the most specialized of those vessels are the lymphatic capillaries. Their job is to collect that fluid and the stuff that is in the fluid and get it into the lymphatic vessels. From there, it feeds into the collecting vessels. The collecting vessels job, and I really don't have room to write it, so I'll sneak it up here on the top. The collecting vessels job is to take that lymph to and from the lymph nodes. So they're going to take that lymph to and from the lymph nodes. Most lymph passes through more than one lymph node. Those are those security guards checking and monitoring everybody's tags and identifications as they're moving to and from in the body. And then these many, many hundreds of collecting vessels will then feed lymph into one of 9 distinct trunks. And do you think we're going to need to identify those 9 trunks and know what part of the body is responsible for collecting, they're responsible for collecting lymph from?
Yes.
Yes, absolutely. And those 9 trunks feed into one of 2 ducts. And it is the ducts that are responsible for then feeding the lymph back into the blood supply. So like I said, this is a one-way path where the lymph, the fluid, enters into the capillaries, becoming lymph, passes from the collecting vessels to and from the lymph nodes into one of 9 trunks, feeding into one of 2 ducts, ultimately being fed back into the blood supply. Your book's got a fun picture that kind of does a nice job of showing this. Not surprisingly, our lymphatic capillaries are located basically in the same place that our blood capillaries are. Here we see a capillary bed in our cardiovascular system. And as we know, that's the structure that is leaky. That is the structure that the extra fluid is coming out of. So it makes sense to have our lymphatic vessels or our lymphatic capillaries interdigitated within them to be able to collect that excess fluid as it comes out. From there, it then travels through our collecting vessels where we see these little enlargements, which are the valves, feeding into a lymphatic vessel, feeding out, of a lymph node, feeding out of a lymph node into another lymph node, feeding out of there. And notice from this, as we mentioned, those collecting vessels feed lymph into and out of our lymph nodes. As such, we will give them names based on their function. A collecting vessel, I'm gonna need a little bit more room, so let's make this a little smaller. A collecting vessel that feeds lymph, oops, did it again, into a lymph node, goes into it. How do you think we would identify such a vessel? What's a term we've used before for going into something?
Reabsorption.
True, but that's really when we're going into and out of a vessel, right? When I'm talking about a directionality of something. Like for instance, sensory information comes into our central nervous system. So when something's going into, what was the term we used for that?
Afferent.
There we go, afferent. So these are afferent vessels. And that would mean the ones that receive lymph from a lymph node. So coming out of it, how would we identify that?
Efferent.
Now notice there is a little bit of a trick to this. This one right here. Can you guys see that line that I just drew?
No.
No, okay, let's make it bigger. This one right here. You guys see that?
No.
You guys don't see me drawing?
Yeah.
Yeah, I can see it.
There it is. This one
There.
right here. Okay. This vessel here, is it afferent or efferent?
Afferent.
Afferent. It is going into the lymph node. What about this one way up here? Is it afferent or efferent?
Afferent.
Which one's this one? Efferent. It is coming out of it. So then what does that say about this vessel here? Is this one afferent or efferent?
Afferent?
Okay, I've had one person say afferent. Anybody want to argue for efferent?
Could it be both, like the top half and the bottom half?
That's not a bad way of thinking of it. We could certainly divide it. That could be one way we could do it. We could put a line and say the part coming out is efferent and the part coming in is afferent. That is definitely one way we could do it. Or like you said at the beginning, it also is both. It could totally be based on which lymph node we're talking about. If we are talking about this lymph node, then it would be efferent. If we were talking about this lymph node, then it would be afferent. So yeah, you could either divide it to the half or it's basically both. It's conditional depending on which lymph node we're talking about. So again, these are directional terms, and that's why I make a point of emphasizing this. It's not like you're going to be able to point to one on that previous picture and say, yes, this is afferent, yes, this is efferent, because it doesn't quite work like that. It's a relative term. But yes, go ahead.
Sorry, did you actually name that? Did I miss the term or?
No, again, it would totally be relevant. It would totally be related to which lymph node we were talking about. If we were talking about this lymph node here, then that vessel coming out of it would be considered efferent.
Oh, got it.
However, instead, if we were talking about this lymph node here, then it would be considered afferent.
Got it. So you would have to specify which one.
Yeah, exactly. And then so it's basically exactly, it's conditional. we would have to, in that case, we would have to give some kind of conditionality to it, so that, because that directional term is ambiguous in this case.
Got it. Thank you.
Yep. But you've absolutely got the right idea. When we are talking about a single lymph node, then it's very clear that, for instance, in this case, these all here are the afferent ones, whereas this one here is the efferent. So while it can be subjective in relative terms, when we're talking about a specific lymph node, then the answer is obvious. All righty. Get rid of all of that. Excellent. So again, we have afferent and efferent collecting vessels, and they feed into the trunks and so on. However, let's talk, before we get to those trunks and identifying them, I want to talk a little bit about these lymphatic capillaries. As I mentioned, the lymphatic capillaries are the most specialized of our lymphatic vessels. And let's actually cheat and go to the whiteboard. This is a very simple illustration, but it's worth taking a moment to do this. When we talked about the capillary beds, we know that there's that vascular shunt that goes between, and then this big elaborate interconnected bed that goes around beneath it. However, our lymphatic capillaries are large, blunt-ended vessels, or let's say thick. And what's special about them is that they are made of overlapping epithelial cells. So, for instance, as we look at the end of it, oops, I don't like that. There's a cell, and 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 there, and there. Oops, I did that one wrong. Nope, that didn't work. There you go, something like that. Oh, this is where I messed it up. Okay. I know I messed it up someplace. You get the idea. All right. So we have these overlapping epithelial cells. And the other key to this structure is only one end of it is anchored to the surrounding tissue. So in this case, this end is connected. This end is connected. This end is connected. So only one side of these epithelial cells are connected to the tissue around them. So let's think about what kind of an effect that would have. I have a big drop of water that comes out of this capillary and doesn't get back in. And of course, as water accumulates in this area, I get an increase of my hydrostatic pressure in the interstitial fluid. And of course, we know that hydrostatic pressure is water pushing against its boundary. And as water pushes against the boundary of this lymphatic capillary, where this epithelial cell is only anchored at one side, what's going to happen?
Water is going to go inside the lymphatic capillary.
Exactly. Basically, this is going to swing this door down. And as a result of that, the water is going to be able to enter into my lymphatic capillary. Of course, now I get an increase in pressure, or in hydrostatic pressure, inside the lymphatic capillary. Let's just say it this way. Lymphatic capillary. As So we get an increase in hydrostatic pressure in the lymphatic capillary, and of course it pushes on its boundaries. And as it pushes on its boundaries, what's going to happen?
It'll push it closed again.
Exactly. It's going to end up pushing the door closed. Right? Like one of those roach motels. They can get in, but they can't get out. Notice that force of the water pushes itself into the capillary and then pushes that door closed. And now, as this lymphatic pressure continues to increase, the water is not going to want to stay here where it's located. So it's going to start moving down the capillary, and it's going to move towards the collecting vessels. So in this fashion, not only can water, but also large stuff, big proteins, cells, right? Things that don't normally get out of the capillary or get into the capillary can easily get into a lymphatic vessel. I said, but there. Water and large stuff can enter into the vessel. So this swinging door makes it easy for things to be able to get in. And again, I appreciate that while this is a simple illustration, my drawing skills are horrible. Here we see again them interdigitated within that capillary. We'll talk about that in a second. Here's the picture that I wanted to show you. Here you get a better job of that overlapping epithelium where just one end is anchored. And so, like I said, this can basically swing open like a door, allowing water and other stuff in. And then as soon as that stuff gets in, it's going to push on the door and slam the door closed again. These lymphatic vessels are found throughout the body. Most places, not all, but most places of the body have lymphatic vessels associated with them. However, as specialized as this capillary is, there is an even more specialized version and that more specialized version of the lymphatic capillary is what is known as a lacteal. Lacteals are these long, straight lymphatic capillaries that penetrate deep into the villi, the finger-like extensions of our small intestine. Our small intestine is where the majority of the absorption of our digestive system goes. And these lacteals are basically responsible for the transportation of lipids and lipid-soluble materials, like vitamins, lipid-soluble vitamins, and things along those lines. So things like carbohydrates, things like proteins, things like nucleic acids, those things go straight into the capillaries, the blood vessel capillary. But lipids and lipid-soluble material instead go into these highly specialized lacteals. All right. Questions on that? Excellent. And here we see some more of these single cell overlapping flaps, these capillaries collecting that lymph and cells and all sorts of other stuff, and then sending it back to those collecting vessels.
Are those just simple squamous epithelial cells?
That is correct. Yep, just simple squamous. Yep. All righty. Excellent. Great question. Any others? All right. From here, as we mentioned, we go into the collecting vessels. the afferent feeding into and the efferent feeding out of our lymph nodes. As I mentioned, these are similar to what we expect in veins. Like veins, they have a tunica intima, they have a tunica media, and a tunica externa. I know our picture from your textbook doesn't do a good job of showing it. It just shows the epithelial tissue. It doesn't show the smooth muscle that would be around it, but it does have that. Like I said, the walls are typically a little thinner than we see in the veins, and definitely more than the capillaries because it has that big, huge, thick muscular wall to it. As I also mentioned, our lymphatic vessels have actually more valves than our veins do. So not surprisingly, they have much more anastomoses. We need to have many, many routes for this lymph to be able to limp its way back to our cardiovascular system. And like I said, the only help that it gets is from that thick muscular wall peristalsis. It's the one thing that it's able to do that the veins cannot. Still able to take advantage of all the help that the veins do, the valves, the skeletal and respiratory pumps, but unlike the veins, our lymphatic vessels can also do peristalsis. We'll talk about the lymph nodes in a moment, but as we know, all these collecting vessels are going to feed into one of 9 trunks. Now, we do need to learn the name of all 9 of these trunks, but the good news is that 4 of these trunks are paired. What does that mean?
There's a right and a left.
Exactly. There is a right and a left of them. The first are the right and left lumbar trunks. And guess where the right and left lumbar trunks drain their blood from?
The lumbar.
True, they're found in the lumbar region, but where do you think they collect their lymph from?
Legs.
The legs, absolutely. From the legs, exactly. We have 2 bronchomediastinal, right? Again, on every test, there's always that alphabet soup term that you're going to have to know, you're going to have to spell at some point, which is such a pain in the butt. But like many of these alphabet soup terms, the name tells you everything about them. Broncho, of course, refers to what?
Lungs.
The lungs. And what's the mediastinum again?
It's that inner space where the heart is located.
Exactly. It's that central cavity within the thoracic cavity where the heart, the aorta, the superior vena cava, things like that are located. So notice the bronchomediastinal, as the name tells you, basically drains lymph from the thoracic cavity. Now, why they didn't just call them thoracic trunks? Well, of course, because anatomists hate you. We have our subclavian trunks, right and left. Where do you think they drain their lymph from?
Arms.
Exactly. We have the right and left jugular trunks. Where do they drain lymph from?
The head.
Head.
Right and left heads? No, of course not. The right side of the head and the left side of the head. Although I make that joke because, as I mentioned, I've been grading my 430 lab exams, and part of the exam, you have to identify this region of the body, and I've had at least a half dozen people write that this is the right cephalic region, like we have a left cephalic region, like we have 2 heads, or the left cervical region or something like that instead of just phallic. So I find that humorous. But no, so obviously, yes, the left side of the head and the right side of the head. So what's left?
Abdominal. cavity.
Exactly. And that is where our single unpaired trunk comes from. Our single unpaired trunk is called the intestinal trunk. And as you have so correctly hit it on the head, its job is to drain the abdominal pelvic cavity. This picture does a nice job of showing all of these. This picture does a really nice job of showing all of these as well. And there's one other structure I want you to see from this as well. Notice here in this picture, and I think the previous picture shows it really nicely as well, are three inferior most trunks, the right and left lumbar and the intestinal trunk. They all feed into this very large, that blue doesn't show up well. Let's see if purple shows up okay. No, that shows up purple as well. All right. I don't care. You get the idea. This big enlargement known as the cisternochylla. The cisternochylla is an enlargement of the lymphatic trunks. And remember, one of the three trunks that is coming to the cisternochylla is that intestinal trunk. Remember that intestinal trunk was fed by all of those lacteals in the small intestine. And so it has a massive amount of lipids and other lipid-soluble material. And do we want to necessarily take all those lipids from those 3 glazed donuts that you had for breakfast and dump them immediately into the cardiovascular system?
No.
No, of course not, right? So the cisternochylla can help to store some of those lipids and even modify some of those lipids before they're deposited back into the cardiovascular system. So this enlargement of cisternochylla is an important structure for storage and even some potential modification of these lipids before they're deposited directly back into the cardiovascular system. So, and again, we see another example of that here as well. Now, when we look at the superior part of this, we can see where we have these 9 trunks that feed into 2 ducts. Now, just doing some simple math there, can 9 trunks evenly divide into 2 into 2 ducts?
No.
No, 9 is not evenly divided by 2. It's not like one of these trunks is going to feed half into one duct and half into the other. So obviously there must be, by math alone, some asymmetry. But it's even more than that. Over here on our illustration, and I will use a dark red to see if I can make it stand out. I need to do that and that. There is a tiny little, I'll actually put a circle around it, the tiniest little duct there. This duct happens to be known as the right lymphatic duct. And if you notice, the right lymphatic duct only receives lymph from 3 trunks. As you can see here, it receives from the right jugular, from the right subclavian, and from the right bronchomediastinal. There you go. So those are the 3 trunks that feed into the right lymphatic duct. So based on that, what are the regions of the body that the right lymphatic duct receives lymph from?
Right side of the head, right arm, and right lung.
Or right side of the thoracic cavity. You got the right idea. Excellent. So that's our first duct. And of course, if we have a right lymphatic duct, what must the other one be called?
the left lymphatic duct.
That would make sense, wouldn't it? But what do we know? Well, anatomists hate us. So instead of being the left lymphatic duct, it is instead called the thoracic duct. Now it's called the thoracic duct because it runs the entire length of the thoracic cavity. If we cheat and go back to the previous picture here, we can see how from the cisterna there is this much, much, much longer duct that travels all the way up the thoracic cavity, looping up here around the top, and then coming in here. So notice again, one spot of my highlighter covers the entire right lymphatic duct, whereas the thoracic duct comes up the entire thoracic cavity. So we can see that big difference there. And we see that big difference then also with the regions of the body that are drained by that thoracic duct. Notice if we identify this, there must be, of course, 6 trunks that are going to feed into our thoracic duct. We can start easy. Well, really, they're all easy because they're all the ones that are left, right? So the left jugular trunk, the left subclavian, trunk, and the left bronchomediastinal. But also both lumbar and the intestinal trunk. So, and I've run out of room here, so I'll cheat and I'll put it up here in the top. When we think of the regions of the body that are drained by the thoracic duct, what's the answer to that?
Legs, abdominal cavity, the left side of the thoracic cavity, left arm, and the left side of the head.
Excellent. Perfect. Notice here, we've done this all with words. But here we have a pretty picture that shows it as well. So notice that the right lymphatic duct drains the right side of the head, the right side of the thoracic cavity, and the right arm. The thoracic duct does all of that for the left side of the head, left thoracic cavity, left arm, but also the entire abdominal pelvic cavity and both legs. And here we have another pretty picture as well. I want to do one more quick thing. I want to come back to this picture. This is actually what I wanted to show you right here. Actually, I'll remove the words so it's not too distracting. Notice also where both of these ducts feed into. Notice that the right lymphatic duct feeds into the right subclavian vein right before it meets the internal jugular and becomes the right brachiocephalic. And notice the same thing happens with the thoracic. The thoracic also feeds into the subclavian, in this case, of course, the left subclavian, right before it meets the internal jugular and becomes the left brachiocephalic vein. So both of them feed their blood back into the veins, back into the subclavian veins, right before they reach the brachiocephalic veins, right before they reach the superior vena cava, right before they reach the right atrium. So that lymph mixes with the blood, oxygen poor, goes to the lungs, gets oxygen rich, and then gets distributed throughout the body. All right, Questions on that? All right, excellent. Like I said, this lymph is basically just excess interstitial fluid. So it's all the things that you would expect to be in the interstitial fluid, all the things you would expect to be inside of our blood plasma: water, ions, lipids, nutrients, waste, plasma proteins, all of those things. But as we also mentioned, our lymphatic vessel, our lymphatic capillaries have those big swinging doors. So larger components can get in here as well. Cellular debris from dead and damaged cells, abnormal cells like cancerous cells or transplanted cells, and then of course harmful pathogens like viruses, like bacteria, like things along those lines. Which is why we don't want to just take this fluid and directly deposit it into the cardiovascular system. We need to pass them through the lymph nodes first. The job of the lymph nodes is to basically filter that lymph before it's returned back to the blood, removing any dead and damaged cells, removing any abnormal cells, or at least setting up an alarm if it sees something that doesn't belong. Now, while lymph nodes can be located pretty much anywhere in the body, and again, this is one of those places where I have to start focusing on my analogies because they don't work quite as well. But normally we're sitting in the classroom together. And the classroom we're sitting in has two doors, one that leads outside and one that leads to the lab tech area. If our job was to protect that room and we had 5 soldiers to do that with, would we evenly distribute those 5 soldiers around the entire circumference of the room? No. You'd put maybe 3 of them by the door to the outside and 2 of them by the door to the inside. You'd cluster them together in areas where we can more centralize and localize our defenses. And that's what happens to our lymph nodes. While a random lymph node can be found all sorts of different places in the body, most of them are clustered together in specific regions. And if we look at that picture we were looking at before, we can see some of the areas where these are occurring. Notice we have our cervical nodes in the neck region associated with that jugular trunk. We have our axillary nodes associated with the arms. We have inguinal and aortic that play a role in what's coming back from our legs and also from our intestinal and other areas like that. So we have these different pelvic, lumbar, inguinal, different clusters helping to provide that defense, clustering these together for that defense. As we talked about, lymph nodes are one of the major places where we find our leukocytes. Remember, in a healthy individual, only about 2% are in circulation. So a massive amount of these are found in our lymph nodes. And not just any are found in here, 2 types in particular. The first are our big eaters, the macrophages. And what type of leukocyte is a macrophage again?
Monocytes.
Exactly. They are monocytes. Remember, monocytes, when they leave the blood, we consider them to be activated. Obviously, if these are in a lymph node, they're not in the blood anymore. And when they're activated, we call them macrophages. Their job is to gobble up anything that doesn't belong. Of course, how do they know that something doesn't belong? How can we tell that there's something in our body that shouldn't be there?
Surface proteins?
Exactly. Remember, we talked about those special surface proteins that we called antigens, right? Now, true, a macrophage may be able to recognize an antigen that doesn't belong, But if only there was a way to plant a flag on an antigen that doesn't belong, that would make it easy for us to, as it waved in the sky, easy for us to be able to recognize it. Do we have flags like that that we can attach to abnormal antigens, foreign antigens?
Antibodies.
Antibodies. And as it turns out, what makes our antibodies are our lymphocytes. Remember in the last section, we said lymphocytes were responsible for our immune response, but we didn't really say how or why. We'll talk more about that in this class. But what we did talk about before is lymphocytes can produce antibodies. And if we can stick that antibody on something that doesn't belong, it's a big waving flag that makes it much, much easier for the macrophage to find and identify something that is abnormal and to deal with it appropriately. So while other types of lymphocytes, other types of leukocytes can be found in our lymph nodes, it houses a massive number of macrophages and a massive number of lymphocytes. All right, we will talk about the anatomy of our lymph node in just, let's leave it on this page, in a minute. And it's a teeny bit early, but this is a good natural stopping point. So let's go ahead and take our first break here. We'll take a 15-minute break. That looks like that means we'll come back at 1:12. And I will start the recording again at that point. Any questions before we take our first break? All right, I'll see you guys in 15 minutes. So, like I said, we're going to talk now about our lymph node anatomy. Most lymph nodes are relatively small. They can be as small as a single millimeter in diameter, up to 25 millimeters in diameter. They are kidney bean in shape, meaning that they are basically convex at one side. So they have an outward curve on one side and concave on the other. This type of shape where it's convex on one side and concave on the other is going to be a common shape that we're going to see anatomically. And when we have this type of shape, the indentation that is formed by the concave surface is what is known as the hilum. And if you notice, the hilum is going to be where the blood vessels and the nerves enter into the lymph node from that region. Notice also we have our afferent and efferent vessels. So let's actually go to our whiteboard and do some drawing of this. Let's get rid of this because that's hideous. And so we have our convex and our concave shape to this. So again, this region here is what is known as the hilum. And as I mentioned, we are going to have our vessels feeding into and out of. Our lymph node. The vessels that enter onto the convex side are the afferent. Whereas the vessels that leave are efferent, exit on the concave side. And notice I didn't just pick these numbers randomly. Do we have an even number of afferent and efferent vessels? These should be the easy questions.
No.
No. There is always more afferent than efferent vessels. Why?
Or collecting ducts and vessels from different regions of the body.
Okay, true. We are taking from more regions of the body and more centralizing everything. That is absolutely true. Absolutely. There's another good reason, though, as well. I always think of it in terms of if I go to San Francisco for the weekend or for whenever, and I'm coming home from San Francisco, it really doesn't matter what time of the day. It could be 4 o'clock in the morning on a Wednesday. I'm going 115 down 80 coming from San Francisco, and then I hit Davis. And what happens when you hit Davis? Any time of the day, any day of the week.
Traffic.
Right, the traffic slows down. Why?
It goes from like 6 lanes to 2.
Exactly. You are starting at, which actually is 114 lanes, and actually going down to 2. You are absolutely correct. And what that does is that causes congestion. That slows things down. Now, remember this lymph node's job. This lymph node's job is to inspect the lymph that's coming to it. So do we want the lymph necessarily rushing through it quickly?
No.
No,
No.
we want it slow. We want it to meander its way through. And so we want to have more time to inspect it. So by having more afferent vessels feeding into it and fewer efferent going out, this causes congestion. And that congestion helps to slow the flow, giving more time for it to be processed. It also doesn't pass directly through, immediately through the lymph node either. In another way that a common organization that we see in many of the structures of our body, we have an outer region and an inner region. When we have that kind of outer region, what do we typically call the outer region?
The medulla.
Well, is the outer region the medulla?
Cortex.
Yeah, the outer region is the cortex. The inner region is the medulla. Oops, I spelled medulla right. The medulla. And we have that type of arrangement with the lymph node as well. Aside from the cortex being outer and the medulla being inner on it, there are some other key distinctions in the cortex as well. One of the biggest things we're going to see in the cortex are these big, large, how do I want to do this? Let's do it this way. These big, large, dense structures, only found in the cortex of the lymph node, not in the medulla. It's the easy way we're going to distinguish them. And these structures are known as the lymphatic follicles or lymphatic nodule. Both are acceptable names for it. And these follicles are the clusters of our lymphocytes. Now, one of the interesting things about these clusters is at the center of them is typically a lighter colored region. This is a very important and very distinct characteristic because it is true for all of these types of lymphatic follicles. They're found in pretty much all of our lymphatic tissues, and they basically look interchangeable. Makes it a teeny bit tricky, but it's also a very distinct structure that is going to be very obvious and easy to see. And these lighter regions at the center are what are actually known as the germinal centers. Now, with a name like that, what does germinal or to germinate mean?
Grow.
Yeah, to grow, to give birth to. Exactly. So think about this. You may not have thought of it in these terms, but you're aware of it. a harmful pathogen comes into a lymph node. Your lymphocyte recognizes it as being harmful, recognizes it as being bad. And what that lymphocyte is going to do is divide hundreds and thousands of times, making plenty of new copies of it. And that's going to occur here in this germinal center. We're going to start making massive numbers of immune cells that are going to help to protect us from that harmful pathogen. Of course, when you get this huge, massive expansion of cells growing inside of this lymph node, what happens to the lymph node?
Swollen?
Yeah, it gets swollen. So often when you have a cold, you feel those swollen lymph nodes in your neck region. If you have little kids, again, we know lymph nodes can be noticed in the axillary region. If you've ever had a little kid before who's had a cold, sometimes they look like huge grapes in their armpits. You can actually feel those big, massive swollen lymph nodes as a result of that swelling from the making of the new cells. So we want that lymph, as it's entering into the lymph node, to pass its way through and around these lymphatic follicles. Now, like many structures, not surprisingly, our lymph node has a big, thick, coated outer shell, fibrous outer shell to it. Any idea what that fibrous outer shell might be referred to as?
Is it the capsule?
Yeah, exactly. It has this fibrous capsule on the outer surface. And one of the interesting things about this fibrous capsule is that there are extensions of that fibrous capsule that penetrate deeply into the cortex. Notice that kind of compartmentalizes the cortex when this occurs. And these projections of the fibrous capsule that enter into the cortex are what are known as the trabecula. Trabecula being singular, trabeculae being plural. And so notice lastly, as that lymph enters into our lymph node, it isn't going to pass immediately through it. Not only do we have more coming in than out, but it has to take this long meandering pathway. For starters, it is going to spread into this space between the fibrous capsule and the lymphatic follicle. And what might a space be called? If only I had a list of the anatomy in front of me, I might be able to figure out what the name of the space between the follicle and the fibrous capsule might be.
It's the subscapular space.
Subcapsular sinus. Notice not, right? Not scapular, subcapsular. The subcapsular sinus. Excellent.
Oh, sorry. My notes were.
No problem. It's
Yeah, the slide was. Yeah.
It is a very common mistake made because they're very similar. Then it goes into this space adjacent to the trabeculae, between the trabeculae and the lymphatic follicle. And what might a space like that be called? Again, you have a study guide to help you to be successful in these sections. There you go, trabecular sinus. Oops. Excellent. So it meanders around. And then we have the medulla. As I mentioned, the medulla does not have any follicles inside of it, but it does have this big, thick, interlocking sinus space. If you remember way back in 430, one of the things we learned about is reticular connective tissue It was one of our loose connective tissues made up of collagen fibers that are elaborately branching. And here in the medulla, these reticular tissues, connective tissues, form what we call the medullary cords. And so then, of course, the space between these medullary cords, what would we call that space? Excellent, the medullary sinus. So after our lymph leaves the trabecular sinus, it weaves its way through the medullary sinus until it ultimately leaves out the efferent vessel. So notice again, we have this congestion, this slowdown, and this long meandering path that it has to pass through. And remember, if something harmful is located in here, our lymphocytes are able to make that tag, that antibody we talked about, that Y-shaped antibody that we can stick on it, that flag that's going to wave and say, hey, that's harmful. And so guess who likes to hang out here in our medullary sinuses?
Macrophages?
This is where the macrophages are. So that those macrophages, when that harmful material comes into here, can recognize that harmful material and gobble it up. So
Macrophages, they also go to the damaged tissue too, right?
true, yes. Again, remember, as we talked about, all of these can be circulating white blood cells that can go to sites of injury or other things like that as well. However, as we said, many of them are housed in these lymph nodes and will stay here for a prolonged period of time as well. So let's look at the pretty picture from your textbook and see these things.
Professor,
Yes.
you said those centers, germinal centers, they divide and produce more lymphocytes.
That is where it's going to occur, yes. When you come in contact with something foreign, that is where the rapid development is going to take place, yes.
So do they contain like hemocytoblasts or the lymphoid stem cells, or just?
Lymphocytes have the ability to divide. So you are correct. They're not stem cells that are in here, but like many of the cells of our body, lymphocytes, remember our white blood cells, we said are true cells. They have a nucleus, they have organelles. And so like most true cells in the body, they are capable of undergoing mitosis. So in an immune response, they will actually divide on their own. We don't need stem cells. They will divide to make their own cells. Yep. All right, if you didn't like my drawing, here is the pretty drawing from your textbook. Again, notice on the outer surface, we have the fibrous capsule on the outside. Extensions of which penetrate deep into the cortex, forming the trabeculi and compartmentalizing our lymph nodes cortex. The cortex, as you can see, is also where we find our lymphatic follicles or lymphatic nodules. Both terms are acceptable. And as we mentioned, one of the key characteristics you can identify about these lymphatic follicles is that they're going to have a light colored center, more cell dense on the outside, less cell dense in the center. And that lighter colored center is what is known as the germinal center. The medulla is distinguished by not having follicles. Instead, they have big, huge medullary cords formed by our reticular fibers, as well as the spaces in between the sinuses. And this is primarily where the macrophages are going to be housed. And again, notice I've done a drawing. We have this drawing here, but we also are absolutely positively responsible for this histologically. So being able to look at this under a microscope is useful. Notice again, we can clearly see that fibrous outer layer, that fibrous capsule. And notice we also see a great example where we can actually see that trabeculi coming in and penetrating deep into the cortex. We know this is the cortex because it is filled with structures like these. These are lymphatic follicles or nodules. Again, you may use those terms interchangeably. And here's the good news. Now that you've seen one lymphatic follicle, you have seen them all. But here's the problem. A lymphatic follicle in the lymph node looks the same as one in your tonsils, looks the same as one in your small intestine, looks the same as one in your spleen. So if they look the same everywhere you find them, how are you going to know what type you're looking at?
Look at their surroundings.
Exactly. You have to look at their surroundings. Notice we've already identified our fibrous capsule in the trabecula. And notice while this illustration, this histology slide, light microscopy slide, doesn't do the best job, notice down here, we can clearly see an area that does not contain follicles, where we can clearly see the cords and the sinuses of the medulla. So we see a very distinct medulla, a very distinct cortex, a very distinct trabecula, a very distinct fibrous capsule. So all of those things tell me very clearly that I am looking at a lymph node. So one of the keys for recognizing your lymphatic tissues is first, see the follicle. Once you have the follicle, you know you're looking at a lymphatic tissue. And then once you have that, you have to look at what's surrounding it to figure out what it is that you're looking at. Anybody print out the histology? Is there anything we forgot about on the lymphatic lymph node that we didn't show?
The macrophages.
True. Okay. So, well, again, remember the macrophages are going to be found here in the medulla. Your book's got a nice illustration. This is a colorized electron microscopy view that shows where some of these things are looking at. I don't expect you to be able to recognize these cells from doing that. But if we look at the medulla in a light microscope, this is what it'll look like. Notice, as we mentioned, you can clearly see these short, elaborately branched reticular fibers made up of collagen protein, which I know you all remember from learning your tissues in 430, so I don't have to remind you of that. But if you had somehow forgotten, first and foremost, of course, shame on you, but secondly, to go back and look at your tissues part of that. And then obviously all these dots are the leukocytes that are hanging out here in this tissue. Now, in looking at these at this magnification, do I expect you to be able to tell which ones are the macrophages and which ones might be lymphocytes or which ones might be something else? No, of course not. Just know that these are leukocytes that we see. And I love this. It's so distinct. Like if you remember back to 430, connective tissues are all so uniquely distinct of each other. This one always reminds me, if you've ever seen one before, a cherry blossom tree. It's very, very reminiscent of what a cherry blossom tree looks like. And that's that reticular connective tissue that we find here in the lymph node.
Did you mention the medullary cords?
So again, the medullary cords are these structures that are formed by those dense reticular fibers or dense packing of reticular fibers here in the medulla that again forms that mesh net-like structure to again slow the lymph down as it's working its way through it. So the cords are the structures and the sinus would be the space in between the cords.
I have a question.
I have an answer.
Are there anything else besides the macrophages that work on filtration?
I'm not sure what you mean by work on filtration.
Like that filter the lymph that
Oh,
comes
yes. Well, obviously the lymphocytes are working that way as well. The lymph is coming in contact with them and they're examining the contents inside there. But yes, all of our white blood cells, there will be eosinophils, there'll be basophils and neutrophils in here, as well as the lymphocytes and the monocytes or macrophages because they're activated. So all of those types of white blood cells would be found in here. It's just lymph nodes have a high congestion of lymphocytes and a high congestion of macrophages, but other ones can be found here as well. Yep. Great question. Any others? All right, excellent. And again, as we talked about, our lymph enters the convex side through the afferent vessels, flows through basically 3 sets of sinuses, right? First, the subcapsular, then the trabecular, and then the medullary sinuses until it finally leaves out the efferent vessels. So we were able to trace that lymph as it passed through those anatomical structures in our description of the anatomy. All right, and one more picture for good measure. Questions on any of that? All right, that is our lymph nodes. However, as we mentioned, those are not our only lymphatic tissues or lymphatic organs. We also have our tonsils. We have, as I mentioned before, what you can write out as a mucosa-associated lymphoid tissue. And once you've written that out once, then you can abbreviate that as MALT on a lab exam or on a lecture exam on an essay question. These include, but are not limited to, structures known as Peyer's patches, and then also our appendix. We also have our spleen, and we also have our thymus. All of these are important lymphatic organs that help to play a role in our immune response. Let's start first by talking about the tonsils. Tonsils are basically lymphoid tissues, which means, again, it's a simple way of saying tissues that contain those lymphatic follicles. And that is crazy huge. And they're basically located around the pharynx. Why? Why might you want to have these tonsils and their defensive cells?
In case you ingest pathogen.
Yeah. And again, ingest could involve eating, passing through the oral cavity, but it also could involve breathing, passing through the nasal cavity. Absolutely. Again, this is one of those places where we want to have our guards clustered together. Most people have 4 tonsils, 4 clusters of these lymphatic tissues in 4 distinct locations. The first of these, as we can see from this illustration, is up at the superior part of the pharynx. The pharynx of course, the muscular tube that is your throat. It is open both to the nasal cavity, the oral cavity, and the larynx. And in fact, that's where it gets the names for the 3 regions. We have the nasopharynx, the oropharynx, and the laryngopharynx. So notice at the very top of the nasopharynx, at the basically internal opening of the nasal cavity, is what is known as the pharyngeal tonsils. These are often what people commonly refer to as their adenoids. So if you've never known anyone who ever had their adenoids removed because of either chronic tonsillitis or even in some cases for sleep apnea, they can be removed. We also have the single lingual tonsils, basically located on the posterior third of the tongue. And then lastly, we have the paired palatine tonsils on the lateral aspects. Let's see how clever I am. Not clever. All right, excellent. Then let's do this. That works. Do they have the other picture too? Yes, perfect. This is what I want. So for those of you who have not taken advantage of it yet, first and foremost, shame on you. So let's go all the way back. Well, like I said, I don't have to say those things for you guys because you guys are 431 students. So you've passed 430, you know how to be successful. And hopefully while you were studying 430, one of the things your 430 instructor showed you was this wonderful site that was put together by Cosumnes River College. Our sister college has a virtual anatomy lab where they've put most of the models and charts in the classroom on there. And what's nice about it is they have two sets of pictures, one of just the image and one with it labeled. I will tell you that we have more models than CRC does, and we are in the process of working on that to make our own. But in the meantime, there's no reason we can't use theirs. We'll cheat because notice they don't have a lymphatic one on here, but since what we're looking at is associated with the respiratory system, we'll come and look at the respiratory one. And notice as we look at this chart does a really excellent job of showing us these tonsils, or at least the pharyngeal tonsil there up at the top, like I said, what is known as the adenoids, and the lingual tonsil right here. It doesn't do quite as good of a job, but if you look very closely, tucked there against the against the lateral wall of the oral cavity is our palatine tonsil. Luckily, though, this particular chart in the classroom has a couple other views. I like this view. This is actually a posterior view of the neck region. So actually, if you look closely at this, you can actually see the pharyngeal tonsil up here near the opening to the nasal cavity. We can see a little bit of the lingual tonsils of the little bit of the tongue that's there, and the teeniest bit of the palatine tonsils. Although, like I said, this is not the best view for these things. However, this is an excellent view. Notice as we look at it here, we can very clearly see this roughened posterior region of the tongue that is the lingual tonsils, right? This is the part that you stick out. This is the part that you use to lick your ice cream cone. This is the part that you use to speak. But the posterior part where it's anchored has the lingual tonsils. And notice these two, change colors here, chewed up bubblegum looking pieces located on the lateral aspects of the oral cavity are the palatine tonsils. In fact, if you look really closely, and I don't remember if you're responsible for this on this exam or if you'll be responsible for it just on the digestive, but I'll go ahead and mention it now. There are two big arches that go from the roof of your mouth inferiorly. The roof of your mouth is the palate. So one goes from the palate towards the tongue. It is the palatoglossal arch. One goes from the palate down towards the throat, the pharynx. So it is the palatopharyngeal arch. And notice in between these two arches is this indentation. This indentation is what is known as the fosse. And inside that fosse is where you find those palatine tonsils. Notice we should have one more good view of the oral cavity here. Oh, they don't have it. They don't have the one straight in the mouth. Oh, well. Nope, they don't. All right, well, so again, Miss River College, what can you expect? But if we look in the oral cavity from the front, you would see those arches, you would see the indentation, and then you would see those pieces of bubble gum that are the palatine tonsils. So here is an excellent illustration that shows us 3 of them. Obviously, this is too inferior of a cut to show the pharyngeal, but unless you have had those surgically removed, you have those 4 tonsils. And again, sometimes they will get removed because of chronic issues of inflammation. However, if you've ever looked inside the mouth of an adolescent, if you have an adolescent, then I encourage you to look inside their mouth. Basically, your palatine tonsils start practically full size, and we kind of grow into them. So when you look inside the mouth of a 4-year-old, they look massive. If you don't have a 4-year-old, just run to the nearest playground, grab some random 4-year-old and look inside their mouth. Don't worry, the parents won't mind. And when you do that, you'll see these huge, massive palatine tonsils. For most people, the size of it isn't an issue, but as I mentioned, in some cases, if they're too severe, they can partially block the airways and can actually cause sleep apnea. So sometimes people will have them removed for those reasons as well. So unless you've had them surgically removed, most people have 4, but I keep using that pesky word most because there are some individuals that have an additional set of tonsils. Notice one more thing this picture shows. This picture shows this small opening here in the posterior part of the nasal cavity. This is the opening of what used to be known as the eustachian tube, which is now what is known as basically it is the tube for the inner ear. Pardon me, the middle ear, helping to equalize the pressure of the middle ear there. Great question. I'll answer that in a second. For this opening for the inner ear, pardon me, middle ear, it is that passageway to help to equalize the pressure. So when your ears pop when you're on a plane or when you're driving up to Tahoe for the weekend and stuff like that, air pressurizes within that auditory tube. Again, used to be called the eustachian tube, auditory tube now. Some individuals will, notice there's a flap of mucous membrane that covers and protects that opening. And some people will actually get a little bit of lymphatic tissue that forms in that region. It is what is known as the small tubal tonsils. Again, there's nothing wrong with having them, except for people who have problems with chronic inflammation, it can block the auditory tube. And if you block the auditory tube, congestion can occur in there, and it can cause chronic ear infection problems. So if you have an infant who has a tremendous number of ear infections, one of the causes can actually be these small tubal tonsils. So the way they resolve that isn't by doing anything to the ear, although sometimes they'll put tubes and stuff like that through the tympanic membrane, but they could also remove those small tubal tonsils, and that can help to resolve it as well.
Could it also cause temporary paralysis to the face? I remember you were mentioning
inflammation of the middle ear. Extreme inflammation of the middle ear can cause that. What we were talking about with Bell's palsy, though, remember that's the maxillary sinus that causes that, not the, pardon me, the mastoid sinus, not so much the auditory tube. But you're right in that the facial nerve does pass through that region, through that middle ear region. So a severe enough middle ear infection could potentially damage that nerve and cause some paralysis, facial paralysis. Many people get them removed when we're adolescent. Again, they tend to be relatively larger in size, and so the swelling of them can become more painful, more discomforting, you know, and so it's not most times when it's removed in adolescence, although it can occur in older individuals as well.
So does having them removed prevent you? I had mine taken out because I had strep a bunch as a kid, and I've never gotten strep since then. So how does it keep you from getting sick by removing? Or is it the swelling The discomfort.
Exactly. It's the swelling. Doesn't mean that you didn't have strep. It just meant that you didn't have the symptoms of strep because you didn't have those tonsils to become swollen and inflamed as a result of that. So
Gotcha.
exactly. So look, if the United States Army were suddenly to fire 1,000 soldiers, we obviously have less defense than we had before, but is it necessarily enough where it's going to potentially harm, or drastically reduce our ability to protect the country? Probably not. And it's the same thing here. Yes, anytime you get those removed, and again, it's one of those things that we've gotten a better understanding of. In my mom's day, it was the thing that everybody did. Every Tuesday, somebody had their tonsils removed and you got to eat ice cream for 2 days. And if you sneezed, you basically got your tonsils removed, mostly because it was done preventatively, because to avoid the pain and discomfort and the problems and stuff. We now have a better understanding of what these structures do. And so it takes more chronic types of issues before we're going to do it. But your immune response is not going to be significantly compromised just because you've had your tonsils removed.
Thank you.
Okay? Yep. All right, we did that. Now, notice I've got a nice low magnification view of a tonsil here. And hopefully the thing that should jump out at you immediately as we look at that is that this is absolutely filthy filled with lymphatic follicles. We clearly see the follicles. We clearly see the germinal center of these follicles, and they are all over the place. So clearly we're looking at some type of lymphatic tissue. So then the trick becomes, what are we looking at? So there are a couple ways that we can distinguish this from a lymph node. Notice the first thing that should stand out is that there's no second region. It's not like these are just clustered on the outside and there's a center part where there's no follicles. The follicles are everywhere. So that should tell us that this is not a lymph node. But we still need to figure out what it is. And so there are two other things that we can look for that will help us. The first is, notice there are these huge, deep invaginations between these lymphatic structures. Big, huge air, really fluid-filled spaces, these cracks, or what are known as the crypts, tonsillar crypts, that are located in there. This is where stuff in the water, stuff in the food, stuff in the air can get trapped and then inspected by these lymphocytes located in this. So those tonsillar crypts are a dead giveaway that we're looking at the tonsil. And there is one more trick. Now, in fairness, we can't really tell from this magnification, but we're smart, sophisticated students, so we should be able to figure this out. These tonsils are either located on the tongue, on the sides of the oral cavity, or the very back roof of the throat. In all of those regions, what type of epithelial tissue should surround the tonsils?
Simple squamous.
Is that what we have in the oral cavity?
Yes.
Non-keratinized.
Okay, getting closer there. Non-keratinized. There we go. Stratified squamous epithelial tissue. Exactly. So if we were at a high enough magnification, should we be able to clearly recognize a non-keratinized stratified squamous epithelial tissue?
Yeah.
Yep. And so if you see this big cluster of lymphatic follicles, maybe you see a deep penetrating invagination, and then on the outer surface of it. And in fact, in some cases, if some of these crypts are big enough, the non-keratinized stratified squamous epithelial tissue will actually line the crypts. So if you see these follicles surrounded by non-keratinized stratified squamous epithelial tissue, then you have your dead giveaways that you are looking at a tonsil. Now, looking at it, can you tell the difference between a lingual and a palatine and a pharyngeal tonsil?
No.
No. Is it possible to tell them apart?
Yes.
Yeah. Am I expecting you to be able to do it?
I hope not.
No, and I'm not. So you just have to recognize that this is a tonsil. You don't have to figure out which of the 4 tonsils it is, but you do histologically need to be able to recognize a tonsil. All right. Questions on that? All right. Let's see what's next. Our tonsils are actually a part of the mucosa-associated lymphoid tissues. Now, based on the name, where do you think these are located? Where do you think a mucosa-associated lymphoid tissue is located?
Mucous membrane.
Yeah, how about associated with mucous membranes? Remind me again, which organ systems are lined with mucous membranes?
The dorsal part of the a distal part of the digestive.
How about all the digestive system? Digestive system, the entire digestive system is lined with a mucous membrane. What other organ systems?
Respiratory.
Respiratory. What else?
Reproductive.
Reproductive. And I can think of one more. At least for boys, it's associated with the reproductive system, but not so for females. There we go. Amy's got it. Urinary. Perfect. All of these organ systems, remember, are open to the outside world. And organ systems that are open to the outside world are lined by mucous membranes. As we work our way through these organ systems, looking at the different tissues, every once in a while, you'll see just a random lymphatic follicle just hanging out there in the middle of the tissue. And any of these single lymphatic follicles located in any of these structures are MALTs, mucosa-associated lymphoid tissues. And we'll run into them all the time. But like the tonsils, there are some areas where these lymphatic follicles are going to be clustered together. Not surprisingly, one of those is in the oral cavity, the beginning of the respiratory and digestive system. And the others are associated with the other end of the digestive system. Like, for instance, Peyer's patches. Peyer's patches are clusters of lymphatic follicles or nodules that are located in the distal part of the small intestine. Now, in fairness, we haven't talked about the digestive system yet, but I'll see what kind of information you guys have. Anybody know what the name of the distal part of the small intestine is called? Like I said, it's okay if you don't. I'm just curious if anybody knows.
Is it the duodenum?
Duodenum is the beginning, the proximal end. So what's the other end called?
The ileum?
Yeah, excellent. The ileum. And what does the ileum connect to?
Is it the jejunum or something like that?
Well, jejunum is the part in the middle. So you're right. The small intestine has 3 parts: duodenum or duodenum, jejunum, ileum. And then what does it connect to? What does the small intestine connect to? The cecum, the beginning of the large intestine. Excellent. And just out of curiosity, what is your large intestine just chock filled with?
Is it microvilli?
True, there are some microvilli in there. But what's actually inside the large intestine? Yeah. Guess what? Every single person in this class, every single person listening to this recording is infected with E. coli. Should we all go running to the doctor because of it?
No, because isn't it technically outside our body if it's
Absolutely.
there?
It is in the large intestine. The lumen of the large intestine, which you are correct is outside of the body. And it's contained within the large intestine, where it actually is helpful and serving some function. Right? However, while it is contained, we also have to worry about it getting free, getting out. So not surprisingly, our Peyer's patches are guards near the end of the small intestine, close to the large intestine, where there's a whole bunch of bacteria, where if they got out of the large intestine could cause some serious issues. So they're going to be clustered together in those areas, making it easy to protect ourselves from things in there. Someone also mentioned the very beginning of our large intestine, this big pouch region here is called the cecum. And notice attached to the cecum is a cute little structure known as the vermiform appendix, or what we sometimes just simply call the appendix. Although I like vermiform. Anyone know what vermiform means? Comes from the root of the same word, vermicelli, or the vermix
Thin.
of the cerebellum. I'm sorry, say again?
The
Thin?
worm.
Yeah, thin, worm-like, exactly. It is a thin worm-like tube. And that thin worm-like tube has a huge bunch of clusters of lymphatic follicles in them. Now, as we all know, the appendix doesn't serve any function at all, right? It's just that vestigial organ, leftover tail or something from when we were birds or dragons or dinosaurs or monkeys or something. Isn't that right?
No.
No. The more and more we've learned about it, the more we've learned that it does have an important function. And one important function is to have these lymphatic nodules that can help to protect us from harmful pathogens, from the bacteria, from our cecum, from our large intestine. But it turns out it's more from that as well. One of the, and again, this was all before the world shut down 2 years ago, but 2 years ago, one of the places where there was a tremendous, massive amount of money being spent on research and effort involved the bacterial flora and fauna of your digestive system. More and more, we're learning that what bacteria are found in your digestive system, in your large intestine, can affect not just your ability to break down food, but they may play a role in intolerances, different types of allergies. They may even play a role in some immune disorders. In fact, if you happen to have just the right combination of bacteria in your large intestine, what can you do as a tremendous money-making opportunity? Come on, I know you know.
Sell your bowel movements.
There you go, sell your poop. Right? Researchers will take it either to do research on, or better yet, they will take your poop, cycle it, spin it down, put it into pellets, and make little pellets that can be suppositories that can be inserted into other people, providing poop transplants. Because if we can change their bacterial flora and fauna, we can change their ability to digest foods, intestinal discomfort, and like I said, it may play a role in some allergies or even some immune disorders. So there's a huge amount of research going into this bacterial flora and fauna. The reason I mention this with the appendix is this long, narrow tube-like structure actually houses many small seed populations of these bacteria. Right? The problem is your large intestine is filled with all these bacteria. But if you eat that gas station sushi, right? And you end up you drinking the water when you're down in Mazatlan, or something else happens that causes you to get that intestinal discomfort, have diarrhea, right? Or you're taking antibiotics because of some other injury or something like that. We can disrupt, those types of things can disrupt the normal population of the bacterial flora and fauna in our large intestine. And these seed populations inside of our appendix can help to store and protect those so that when we finally pass that gestation sushi, we can use these seed populations to repopulate the bacterial flora and fauna of our large intestine. So our vermiform appendix is pretty darn important. But at the same time, the same way this shape helps with the bacteria, this shape is what causes the problems as well. Because as we've just finished talking about, as we know, if a harmful pathogen comes in contact with these follicles in my appendix, they're going to make many new lymphocytes, and our appendix is going to swell in that region. And when it swells, it could block up the opening. And if it blocks off the opening, we can start to get an accumulation of fluid inside this appendix. And what do we call that condition?
Appendicitis.
Appendicitis, absolutely. Appendicitis can be very painful, very uncomfortable. And the other risk you have is if it pops like a balloon, you're basically rupturing a hole in your large intestine, letting that feces, or soon to be feces, letting all that bacteria into your peritoneal cavity. And is that where we want bacteria and feces?
No.
No, it can cause major, major issues, and in extreme cases can even cause death. All right, so there's the good and the bad of the appendix. I'm not going to hold you responsible for the appendix histologically, but I will hold you responsible for the Peyer's patches histologically. Again, notice as we look at this very clearly, we have this dark, dense follicle. Notice I don't see a germinal center. Does that mean Peyer's patches don't have germinal centers?
No.
No. Then why don't I see it?
Are they located around the surrounding area or?
Yeah, look, remember, histology is a two-dimensional view at a three-dimensional structure. If I throw an apple into the air, close my eyes, and just randomly take a slice through it, as I look at that slice piece, am I always going to see the core?
No.
No. Does that mean that that apple didn't have a core?
No.
No, it just means that the cut I took of it didn't show the core. And that's all that happened here. This is probably a more lateral view of this particular Peyer's patch. And so we didn't cut through the core. We didn't cut through the germinal center. But we still have that congestion of cells. And again, if it had, and sure on the exam, I would have one that has, this is the picture out of your textbook, so that's why I just used that. But obviously on an exam, I would give you one that had a nice obvious germinal center. But with that germinal center, is there anything about this lymph node that makes it look like, this lymphatic follicle, that makes it look different from any other lymphatical follicle? No. So again, we have to look at the surroundings. Here in the small intestine, we are going to see these intestinal crypts, these invaginations of the tissue filled with goblet cells to produce mucus. We'll see those villi that are extend out to give those finger-like extensions that are going to increase absorption. And what type of epithelial tissue lines the small intestine?
Simple columnar.
Simple columnar. So by looking at the surrounding tissue, the surrounding structures is how we're going to be able to tell what it is that we are looking at. Whether it's a lymph node, whether it's a tonsil, whether it's a Peyer's patch.
Professor, quick question.
Yes.
So it would be simple columnar, but it wouldn't be ciliated columnar because they have a villi, right?
Yes, they would have microvilli, but they would not have cilia.
Okay.
But yeah, this view, you're not going to see the cilia, but if we were at a higher, closer magnification, pardon me, the microvilli, but at a higher magnification, you would certainly see the cilia. I did it again. Certainly see the microvilli. But yes, remember cilia just in the nasal cavity, just in the trachea, right? They're ciliated pseudostratified columnar.
Right.
So simple
Thank you.
columnar will just have microvilli. There, and I actually said it right that time. All right, two more. Next is the spleen. The spleen is over here on the lateral aspect of the abdominal pelvic cavity, opposite of the liver. Obviously, it's not the same size as the liver. As we know, the liver is the largest visceral organ in the body. But the spleen is pretty big. Here we see another nice view of it with a transverse section. Here, of course, is the liver. Here, of course, is the stomach. And then over from that is the spleen. The spleen is the largest lymphoid organ in an adult. What does that mean?
There's a bigger lymphoid organ in children.
Exactly. That is exactly what it means. The spleen is a very well vascularized and very delicate organ. I make a point of emphasizing this because when someone is, for instance, I don't know, riding a horse or playing a sport or takes a severe fall off of a skateboard and damages their spleen, typically when that happens, the spleen has to be removed. Because it is so well vascularized and so delicate, it's not the kind of organ where we can just sew it back together and then cross our fingers and hope for the best. So if the injury is severe enough to the spleen, in most cases, they will just remove it rather than trying to repair it in any form. And with that massive blood supply, not surprisingly, its primary function is to filter the blood. Basically, you can almost think of it as the lymph node of the blood. The same way lymph comes to a lymph node to be filtered, to be processed, blood comes to the spleen to be filtered, to be processed. Now, some of that filtering involves the breaking down and recycling of our red blood cells. It's what gives the spleen a very dark, deep, reddish, purplish color because it stores a tremendous amount of iron from a tremendous amount of hemoglobin that is broken down from our old red blood cells. But it is also going to examine the plasma the same way that the lymph node examines the lymph, looking for abnormal cells, looking for cancer cells, looking for dead, damaged cells, looking for pathogens. And if it finds those things, just like the lymph node, it can stimulate an immune response. Notice, not surprisingly, with two main functions, there are two main components to our spleen. Notice as we look at a light microscopy view of our spleen, like a lymph node, like many of the organs, the kidney and plenty of other stuff as well, there is a fibrous capsule on the outer surface. There can actually be some trabeculi that penetrate into it, and we see some of those here as well. But it doesn't compartmentalize it the same way that the lymph node does. Notice also that our follicles are spread out. They're not clustered together the same way they are in a lymph node. They're much more spread out and diverse. And when that occurs, that basically gives two regions to the spleen. We have this middle area that is known as the red pulp. Remember, color is one of those things in histology that doesn't matter except when it does. And this is one of the times when it does. Why do you think the red pulp is called the red pulp?
Because it's red.
Because it's red. And why is it red?
Because of iron.
Yeah, exactly. Because of the stored iron from the breakdown of the red blood cells. Whereas, where our white blood cells are housed in these, what we would call splenic follicles, form what we call the white pulp, because this is where the white blood cells are located. Now, remember I said all lymphatic follicles look like all other lymphatic follicles? Well, like any good anatomist, I lie. And this is another example of where that has occurred. Because if you look closely, notice in this one in particular, this one is the best example of this. Remember, in this case, our splenic follicle's job is to monitor conditions of the blood. And if you're going to monitor conditions of a blood, you need a blood vessel to go through it. So going through the center of our splenic follicles are going to be what are known as central arteries. Notice here we see a central artery. This one we can see entering in through the side of our follicle. And if you look really closely, here you can actually see a cross section through that splenic artery as it's passing through. Now notice, do you see the splenic artery on this one?
No.
No. Does that mean it doesn't have a splenic artery?
Nope.
No, it just means that this particular one section through this spleen doesn't show it in this one. But here, this is where you see it the best. These longitudinal views of that central artery are a dead giveaway. You are looking at a spleen. Now, again, it's called red pulp and white pulp because, as we mentioned, the red contains the hemoglobin, contains the iron, which is going to store oxygen and give it a reddish color. The white blood cells don't have that, so they're lighter in color, they're white. However, when we use different stains, can that color change? Yeah, notice with this particular stain, it's not as easy to see the red coloration of the red pulp. But notice as we look closely at it, we can still see all those characteristics that are going to tell us, well, almost all the characteristics that are going to tell us this is a spleen. We have a fibrous capsule. Here we see 1, we see 2, we see 3 spread out splenic follicles, or what we could refer to as the white pulp. Everything else is the red pulp. And while it's not the best example, again, I wouldn't use this on the exam, but if you look closely, there's that little bit of red right there. That little bit of red right there is actually the central artery that is passing through that splenic follicle. Again, this one isn't good enough where I would use it on an exam, but it was one that I found as I was quickly looking for these things. I just wanted to show you the red pulp's not always going to be red. So don't get fooled by that. But the splenic follicles are the white pulp. Everything else is the red pulp. All right.
So the blood enters to the spleen through those arteries, and how does it leave? Where are the veins located?
Great question. So it's one of those great examples, as we've talked about before. Obviously, a blood vessel coming in has to feed into a capillary and has to feed out. So as we go back to that previous one, while it is that previous picture, while it is indeed called the splenic artery, that is the name for it. Is it always necessarily an artery? Could it be a vein coming out of it? Yes, but the name they give of that blood vessel found in the center, we always call it a central artery. But it is going to, you're right, it is going to feed into veins that are then going to drain out of the spleen as well. Yep. All righty. one more organ we need to talk about, and that is the, how are we on time? We're doing great. The thymus. The thymus is this two-lobed organ that basically drapes over the top of the heart. Notice this is in a mature adult. In an adolescent, this can basically drape over the entire surface of the heart. So this is that lymphatic organ that is largest and most active during childhood. And again, when I say largest, I mean relative size. Is the thymus of a 1-year-old going to physically be bigger than your thymus?
That'd be crazy.
Yeah, of course not. No, yours is going to be much, much bigger, right? However, your body is also much, much bigger as well. So we're talking about relative size. In an adolescent, its relative size is by far much, much larger in relation to that as an adult. And in fact, it peaks in size around age 2, at which point we continue to grow and the thymus doesn't. Then you start to reach that magical age of about 35. And around 35, it slowly starts to shrink in size. And by the time you reach that magical age of 50, it's pretty much undistinguishable from the connective tissue and adipose that surrounds the heart as well. That doesn't mean it's gone away entirely. There's still going to be some of these individual little lobules. These small little compartments are the lobules. There'll still be some lobules left. It still will be able to function, but it's not nearly as large and not nearly as active as we age, as we get older. The thymus is a lymphatic organ, but it's also an endocrine organ. It produces a hormone called thymosin. And thymosin's job is to mature lymphocytes. And just out of curiosity, a lymphocyte that matures here in the T thymus using the hormone T thymosin, what do you think we call lymphocytes that mature in this region, the thymus, with the hormone thymosin?
T cells?
Exactly. This is where we get our T cells. Our T cells play a huge, important role in our immune response, which we'll start talking about, I think, on Tuesday. Yeah, Tuesday we'll start talking about our immune response and our body defenses. So we'll start talking about that on the Tuesday. And so, yeah, this is where we make our T cells. This is where we start making our specific defenses to protect us from foreign pathogens. This is where they mature, where that lymphocyte becomes capable of providing a defense. And of course, we have a fancy word for that. We say it becomes immunocompetent, capable of protecting us. It's like a soldier. You take that high school senior who graduates wet behind the ears, you send him to the military, and he goes through basic training. When he comes out of basic training, he has now been trained as a killer. But has he ever killed anybody?
Nope.
No,
No.
probably not. Hopefully not, right? He hasn't killed anybody while he was in basic training. Absolutely, right? So he's become capable. He's learned how to, but he hasn't done it yet. And that's what we mean here. Immunocompetent means the cell is ready to fight, but it hasn't fought yet. It's still naive. It still hasn't been in a battle yet, but it's ready. And that's the goal of our thymus. Notice your thymus is not on your histology list. I'm not going to hold you responsible for identifying it histologically. So you don't have to worry about that. You just need to know what it does. All right. Questions on that? All right. So here's the game plan. We are going to go ahead and take one more break. When we come back from this break, we will then go over the instructions for the group presentations. There is a handout in the modules that I will release. Actually, it's already available now. If you want to look at it during the break, you're welcome to do that, but we will go over it together. Once we go over it together, I will answer any questions that you guys have. After that, we will then, so we'll go over together. Once we go over together, I will then, using Zoom, randomly form groups. Once you've got your group, you will then pick your topics. And with, oops, if I spelled topics right. And with that, you will then use the rest of the lab time to start your work. I will provide you some lab time today. I will provide you some lab time on Tuesday to work in class time to work on these, but I will tell you right now, it is not going to be enough. So to help you, I will be setting up group discussion boards. Those group discussion boards will allow you to communicate asynchronously. Again, I know that not everybody is necessarily going to be available at the same times, but if you have the means, if your schedules are compatible, then I do encourage you, you can actually meet in Zoom, you can meet in the open lab, you can meet anywhere you want if you are, again, comfortable doing that, but it's not required. No one is required to physically meet with the other people in the class. Between the time we have in the classroom and your group discussion boards, or you can text or you can email, it is perfectly acceptable for if me, Aravek, and Noga are in a group together, we can decide I'm doing A, B, and C, and you're doing D, E, and F, and she's doing G, F, and Y. And then we can do our parts, put them together, and again, do it all asynchronously. It's easier if you meet, it's funner if you meet, but it's not required. So again, you've got your group discussion boards, you can share email addresses, use your school email if you don't want to use your personal ones, but so to do this, but you will work together in groups on this, and we'll get 1 grade as a group for this as well. But you're welcome if you want to, and if you're comfortable to meet outside, you're welcome to do that. Or like I said, in Zoom, or go to the open labs or anything like that, but it is not required. All right, let's go ahead and take our break. It looks like it is 2:30. So let's come back, 2:28, but we'll call it 2:30. Let's come back at 2:45. And at 2:45, we will start and we will go over the group presentation then. All right, I'm going to go ahead and pause this, and I'll see you guys in 15 minutes. Let's go ahead and get started and talk about our group presentations. So as I mentioned, today you will be randomly assigned into a group. I can randomly do breakout rooms, and that you'll be doing. This will be your group, and you are going to work on a presentation that is going to be 8 to 12 minutes. You will have to be here today. Obviously, you're out here today, so that's not an issue. We won't deal with any of that. Again, there looks like there's 25 of us, so it looks like we'll get a couple groups of 2, and the rest will be groups of 3 that we will have on that. Again, I will set up a discussion board for you, just for your group, so you can share information, you can share communication. When I set up the group for you, what I would like you to do is someone please make sure you put everyone's names, and more importantly, the topic that you guys have in your discussion board, so that it is easily accessible and easy for me to see, so that you can do that way. Like I said, you will have time today in class to work on this, and you'll also have some time in the next class. So if you're having problems, I can be here to help you, but that won't be enough to complete this activity. As I mentioned, the other big thing you're going to be doing today is picking your topic. You're going to pick your topic from one of 3 categories. You are either going to describe to us a disorder of the lymphatic system, an autoimmune disorder or an immune deficiency. Now, what do I mean by that? What is an autoimmune disorder?
Like Crohn's disease?
Okay, that's an example, but what's the definition of an autoimmune disorder?
Where your body attacks its own body tissue.
Right, exactly. Autoimmune is where our own immune system attacks some portion of our body. Remember, we've already kind of hinted at we need to be able to distinguish what's us from what's not us. And sometimes we have a problem with that and we incorrectly target ourselves. And so that is an example of an autoimmune disorder. How is that different from an immune deficiency?
Not
Maybe
having- Go ahead.
Sorry, I was just gonna say a deficiency is like your body not making enough.
Sure, you either don't make enough, or what you make is non-functional, or you're lacking something altogether. Exactly. So maybe you don't, and again, I don't even know if this is a real disease, but let's say, for instance, you didn't make eosinophils, right? That would be an immune deficiency where you didn't have or lacking some component or didn't make enough of it or something like that. So when you pick your topic and you're presenting it, make sure you let us know which of these three categories it falls into when you're doing your presentation. What you're going to be doing in your groups, as you can see here, is when I put you in into your groups, and actually here, let's write this here. Once in your groups, I want you to pick 4 topics and put them in order of your preference. Once you have 4 possible topics and you've put them in order in preference, then one student and one student only will come back to the main room. You're going to all be in breakout rooms. And so one student only will come back to the main room. And then in order of return.
It's hard for me to hear you.
I'm sorry? I didn't hear what you said.
Your voice was cutting out with me.
Oh, okay. I'm sorry. Well, here, the window's open behind me. Maybe that makes a difference. All righty. So then you'll come back in an order of the return. You will present me with your list. I will try to have the waiting room set up, but honestly, if you're in breakout groups, I don't know if I can have the weight room set up. So I'll try, because it's easier for me to keep people in order in the weight room. But if not, then we'll just do it in still the order that people go. You'll present your list. And again, then if someone hasn't taken one of your options, you will get your pick. Again, there's going to be no duplicates. So each group needs its own unique topic. We're going to have somewhere on the order of, 9 groups or something like that. So there's no reason. There's plenty of disorders out there. So everybody will have their own. I don't want 2 groups doing the same topic. So that will not be the case. And again, if on the off chance someone has picked all 4 of those already, then you'll go back to your group and you will you'll go back to your group and you will pick another one until everybody at the end of today will have a topic that they will be doing their project on. Okay.
Is there a list of disorder we choose, or we just look up, or?
There is this thing called the almighty Google, which is but all kidding aside, your textbook has some that are listed, and then you'd be surprised at the number of people who know someone or have someone that may have a disorder, so that it may be of interest to them that way. Or you can just, like I said, you can do a web search on your phone for autoimmune disorders or immune deficiencies. If a group is really stuck, I'm happy to provide some ideas for topics, but usually between your group and the ability to even a limited search, you should be able to come up with plenty of potential topics that you would be interested in exploring. All right. There are research guidelines that we'll talk about, and you'll see in just a minute on the next page. You are going to, your textbook can definitely be one of the references that you use for this, but you must have a minimum of at least 3 references. So you definitely need to go outside of your textbook. Those references could be articles, newspaper articles, journal articles, websites, reputable websites, right? None of this AON stuff, but reputable websites that you're looking for things along those lines. And make sure you give us on your outline at the end your references. I don't care about the format. It doesn't have to be APA format or anything along those lines. What I want from your references, if somebody is interested in the topic you discussed, they can easily find it. So make sure your references are on the outline. Again, this is a 50-point project. And if you do not put your references, then you will lose 50 points off of your total for this presentation, which takes it down to a 90%. So it is very important that you do them, and that also that you turn your outline in on time. As I mentioned, you are doing a live oral presentation using a slide, using slides. You can use any of the formats if you want to use Google Slides or PowerPoint or Prezi or however you want to do it. If you want to make a PDF and show that page by page, you are welcome to do that. What I ask for is 3 things. Obviously, you need to have some type of slides. And like I'm doing, you will share those. So you will be sharing the screen, you will be presenting that. So notice also, like me, you will only be a little box up in the corner. However, you do need to be on camera. You need to be on camera, and your whole group, let's say it this way. Needs to be on camera. I would encourage you to just have one student that has the entire presentation and is presenting them and clicks through the slides. Again, if you really, really, really want to have each of the 3 of you make your own slide presentations and present your 3 parts, you're welcome to do that. But switching permission to share the screen, bringing up the new slides, doing all that stuff takes time, and it does count towards your 8 to 12 minutes. So technical issues, trying to run a video, switching between students, all of that counts against your time. And I know 8 to 12 minutes sounds like a lot, but trust me, it goes fast. So I strongly, strongly encourage you to just have one student have the entire presentation. And they can share it like I'm sharing it right now. But everybody is going to be on camera and everybody should present. However, that being said, you are not being graded on your presentation skills. If you want to have your entire speech written on a piece of paper and you want to stare at it and read it the whole time you are presenting, as long as you're on camera, that's fine. I don't care. Just make sure that people can hear what you're saying and you're on camera. No one's going to be looking at you anyway. They're all going to be looking at the slides you put up on the screen. And you're not being graded on your presentation skill. You are being graded on the information only. So again, this is as low pressure of a presentation as you will ever give in your academic career. Also, with a slide presentation, like I do in class, I do not want your script on the slide. You can have a script that you read, but that's not what should be on the screen, because you want people listening to you. You don't want people reading what's on the screen. So what's on the screen should be an outline or the pictures that you're going to be presenting. In fact, as I mentioned, you need to make an outline, not a script. And like I do for class, if you want to take your slides and make your slides your outline, that's perfectly acceptable. So the same way that I post the slides, and it's not everything I say, but it shows you the important information and it's the breadcrumbs that we follow in lecture, then you are absolutely positively welcome to do that as well. All right. One other important point on this, I will not be recording the presentations. These presentations are required. You need to participate in it to get this grade, but I appreciate that not everybody is necessarily as comfortable as I am being on camera and being recorded. So I will not be recording the groups to take that pressure off of the groups that are presenting. However, that means those of you who aren't presenting during that time need to be extra vigilant in taking notes and paying attention. I know it's very easy while someone else is presenting to be in my group chat on my phone, talking to my rest of my group and not paying attention to what people are saying because I'm worried about the fact that I have to go next. Don't do that, because then you're going to be missing the information that the group before you is presenting. I expect you to all be active listeners. And at the end of each presentation, I expect you to ask questions. Because here's what's going to happen. We are going to have a question and answer at the end of the presentation. It does not count against your time, but we will have a question and answer. And one of two things are going to happen. Either the other students in the class are going to ask you questions about your presentation, or I'm going to ask you questions about your presentation. If nobody asks you questions, then I will. And who would you rather have ask you questions? Somebody else in the class or me? Me? Well, I appreciate that. You want to be asked questions from the other students, absolutely. So please make sure you ask questions of the other students and then they will ask questions of you. All right? Tit for tat that way. All right. It is important to provide the information so that we all can understand the basic concepts. But it is also equally important to be precise in what you say. The presentations are going to be from 8 to 12 minutes. If you do not talk for 8 minutes, your group will lose points. However, if you go over 12 minutes, you will also lose points. Now, typically when we hit about the 10-minute mark, I will try to give you a quick indication that you have, like in the chat window, I'll type that you have 2 minutes left to give you a warning. You should be practicing these presentations to have your timing down, but I know sometimes when we're actually doing it for real, we get excited and talk too fast, or we get excited and get flustered and it takes a little bit longer. So prepare for that. But if you go over 12 minutes or you're under 8 minutes, you will lose some points. So practice. Time yourself, pay attention to those things while you're doing the presentations. What else? As I mentioned, each group is going to present an outline. Remember, I've got it here, do submit it online. And it's not the night before the presentation in this case, because we have a Tuesday presentation. So it is the Friday before the weekend. So Friday, October 1st at 6 p.m. is when your outlines are due. I'll get them posted online by 8 o'clock that evening. and then you will have the weekend to look ahead at the materials that people are going to be presenting. Failing to turn in your outline on time will be a 10-point deduction of your total score. And I do mean on time. If you turn in your excuse me. If you turn in your outline at 6:05 p.m., it is late, and you're losing 10 out of your 50 points. There is no reason for this to be late. I know it's awkward having something due on Friday, so guess what? Finish it on Wednesday and turn it in before your Wednesday class so you don't forget. But if you need that extra time, I want students to have that extra time, but I want to have these up for the weekend. So it is vitally, vitally important that you complete them and turn them in on time. All right. Also, make sure you relate the information to our class. Oops, helps if I spell relate correctly. If your disease involves the skin, then remind us of the things we talked about when we talked about the integumentary system. If it involves the heart, remind us of the things we talked about 2 weeks ago about how the heart normally functions. It gets a little bit trickier if yours involves like the digestive system or the respiratory system or something we haven't talked about yet. But you can say, hey, when we get to the digestive system, we're going to learn that the stomach normally does this. And my disease causes it to do this. Right? So even if we haven't covered the material yet, you can still easily relate it to the class so that, again, in the class, you're reminding us of the normal function, and then you're telling us how that normal function gets disrupted. Like I said, I expect you to be active in the learning process. This information you are going to be responsible for on the lab and the lecture exam. Now, by that, do I mean that you have to know that disease X is treated by medicine Y that is given 3 times a day, with a meal? No, of course not. But do you need to know the general characteristics, the type of disorder it is, right? The type of symptoms, how those symptoms are treated, general information like that? Yes, absolutely. And like I said, I will not be recording these, so you need to be active in the learning process during the presentations. And like I said, at the end of the presentations, not counting towards your time, will be a quick opportunity for students to ask questions. So make sure not only, and again, that helps you to make sure you understand the information. And by you asking them questions, you are certain that I will not. Lastly, and most importantly, you want to know what you can do to be successful on this. Well, right here in part 11, I have provided you with exactly how you are going to receive your 50 points. You are going to be responsible for describing the disease and giving us the physiological basis of the disease. And if you do that sufficiently, you will get 14 points. You are going to tell me how the disease is detected, what the symptoms of the disease are. If you do that thoroughly, you'll get 14 points. You're going to tell me the risk factors, how the disease is treated, if the disease is treated. As we go through these, some of these you'll learn there is no treatment for these diseases. In those cases, we can't really fix the disease, but what we can do is treat the symptoms. So even if you can't fix the disease, make the disease go away, there are ways that we treat the symptoms. And so you'll provide us with that information. You do that, you'll get 10 points. Make sure you relate the material to the class, either 430 or 431. You'll get 6 points for that. And again, for having a good slide presentation, again, I'm not grading you on the style, but if all you have is a script up there with a bunch of words, that's not really indicative information. If you've got a disease that causes a horn to grow out of the side of their head, then you should have a picture of somebody with a horn growing out of their head, right? So as long as you're using the appropriate visual aids to summarize the information, and again, that can be anything. If you've picked your disease because your grandma has it and you want to show us a picture of your grandma, say, hey, there's my grandma, she has the disease, right? Some type of visual components, not just words on your slides. You will get 6 points for that. And it's just that simple. Now, does that mean you have to exactly go through this order of how you're presenting information? No, but these are the things that I'm looking for. You can present it in any order that you want. Just make sure that these components are all there, and that is how you're going to get your 50 points. All right. Questions on that? Really? Nobody has any questions? All right. Then,
Yeah.
yes, go ahead.
So I guess I'm just a little confused on what. So it says one student only come back to the main room, and in order of return, present list.
Okay, so
I don't know
let's say your group comes up with a topic and your 4 topics, and Aravic comes up with her 4 topics as well. And let's say her top item is uncontrollable breakdancing, and your top disease is uncontrollable breakdancing. If she comes back to the group first, to the main room, because again, I'm going to put you in breakout rooms. So one person is going to leave the breakout room and come back to the main room. When you come back to the main room, if Aravic comes back first, she's going to say, we would like to do uncontrolled breakdancing. And I'll say, great, no one's picked that yet. That's yours. Go work on that with your group, and I'll send her back to there. You came second. You can't do uncontrollable breakdancing anymore. But whatever your second disorder is, then you would get that.
Hop and lockitis.
Exactly. And as long as it's different enough, then it should be fine. All right. Did that sufficiently answer your question?
Yeah, thank you.
Excellent. All right. Great question, Claudia. You do not make the discussion boards. What's going to happen is, like I said, Zoom has this nice program where I can set up breakout rooms, and I can just randomly assign you. And that's exactly what I'm going to do. It's going to randomly assign you into a group, and then you guys are going to go into your breakout rooms. Once I have that information, I am then going to take those lists of names and go into Canvas and make discussion boards specifically for the groups. So we will have a group presentation groups, and each group gets their own discussion board. So I will be setting that up. So you don't have to do anything. After I get all the topics from everybody, it'll probably take me an hour or so. So if not by the end of class, then by the end of today, you will have discussion boards set up in Canvas. So when you go to the discussion board, there will be one that is set up specifically for you. So yes, whatever topics you are picking, whatever disorders you are picking, that information that is presented by you and by the other groups could appear on both the lab and lecture exam. Like I said, if one of these disorders causes a big horn to grow out of your head, could I show a picture of somebody with a horn growing out of their head and say, hey, what disorder is this on the lab exam? Yeah, absolutely. Or I could, as an essay question, right, describe a disorder that would cause, a horn to grow out of your head, or what are the symptoms of, disease X, or how do you, or what are some of the risk factors of disease Y, or multiple choice questions on it, or things like that. Absolutely. This is information that could be on, will be on the lab and lecture exam. Yep.
When we come back to the main room with our topics, is that I'm trying to remember the breakout rooms from last semester. Do we have to just leave the Zoom and come back in, or is
No,
there a
you
way
don't-
to
great
do it?
question. There is a way, when you're in the breakout room, there's a way to leave the breakout room, and that sends you back to the main room.
Perfect.
So
Thank you.
don't exit Zoom. Although if you do exit Zoom, you can then get back in that way as well. But you should be able to just leave the breakout room and come back directly into the main room. All right.
Okay, and then are we gonna meet with our group today?
That's what you're doing right now. So what we are doing right now, so let me see if I can do that and do that. So yes, so here's what's going to happen. I'm going to put you in your groups in just a moment. Actually, I had just started. Where did it go?
And we're picking it today, right?
Yes. So yes. Okay. So here's what's going to happen. You are going to be moved into your breakout rooms. Once you're moved into your breakout rooms, you are then going to pick your topic. One person will come back. I will tell you which one of your topics you want you are getting to do. You will then go back to your group, tell them which topic you guys got, and then you can start game planning, saying, you know, and again, you've got the list. You don't have to do it this way, but you have the list right here in front of you. So I could say, hey, I'm going to describe the disease and do the physiological basis. Hey, Amanda, how about you do how the disease is detected in the symptoms? And Aravek, why don't you do the risk factors and how the disease is treated, right? So you can divide it up however you want. And you guys can come up with a game plan, and you can talk about whether you're going to meet, or you can just exchange phone numbers for texting, or do it on Zoom, or, hey, I go to the open labs. Do you go to the open lab on Friday? Do you want to meet on Friday? Whatever it is, you have the rest of the class time today, which is a little over an hour, to kind of game plan and decide what it is that how you're going to attack this and be successful with it. All right.
Okay.
Excellent. Any other questions? All right, I'm going to then go ahead and open up the rooms for you. You guys can go into your breakout rooms, pick your topics, and then come back here when you are ready. Let me stop sharing that. And then, all right, away you go. Hey, Dina. Dina, are you there?
Hi.
Excellent.
Sorry, I want to come back here. I actually accidentally left the room altogether.
No problem. So what do you guys want? You got
We
first pick.
were going for autoimmune disorders,
Okay, which one?
and we have the list of 4. The first one being Addison disease.
Yours.
Perfect. And if I understand correctly, we don't have to give you all 4. It's 1, correct?
Right.
Okay.
As more people pick, like if the next person comes, you're first, so you get your first pick.
Gotcha.
But as the next person comes, they need to have at least 2 and so on and so forth.
All
All
right.
right, which group are you in?
That's a great question. I'm in the group with Jacob and Tiffany. I'm afraid I didn't get the number.
Okay, let me find, there you go.
You
You're,
said
looks
the
like you're in room
different-
2, so I will send you back to that.
All right. Thank you.
All righty, there you go. Hey, Nathaniel. What do you got for me?
Hi, Professor. So we chose, our first one was lupus.
Yours, done.
Awesome. Also, I apologize. When I was leaving the group, I accidentally clicked leave meeting, so I had to go all the way back through the system.
Well, and yet you were still the second one here. So you got your disorder. Which room were you in?
I believe 6.
6. So that's with Maximilian and Ryan.
Yes.
All right, I will send you back. Congratulations, you're all set.
Awesome. And then we can just talk through it and
Yep, figure out what you're doing with lupus.
okay. And if we all just kind of want to split, then we can leave.
When you're ready, you're welcome to go.
Awesome.
Yep. Hey, Catherine. Hey, Catherine, what do you got for me? Nope, now I can hear you.
Okay, there we go. So we chose HIV.
Okay, excellent. I love that. Now, here's the one thing I will tell you with that.
Okay.
The one difference between your group and everybody else's group is some of these groups with their topics are going to have a hard time finding 8 minutes worth of material to talk about, right? You got the opposite. There is so much information out there. You guys are going to have a hard time trimming it down to stay under 12. Okay.
Okay.
The other thing that I want to make sure you emphasize, because I don't think enough people understand and appreciate it, is HIV and AIDS are not the same thing. Right? Obviously they're related, but make sure you explain the difference between HIV and AIDS. All right. I love that you guys are doing that. That's awesome. What group are you in?
I am with Ala and I can't remember the other guy's
Sean?
name. Yes.
Sean. All right, group 1. All right, I'm sending you back to your room then.
Okay.
All right, bye. Hey, Noga. You there? You're muted.
Okay.
Excellent.
Our first choice is rheumatoid arthritis.
Done.
Cool. Okay.
Perfect. Excellent. All righty. Perfect. So what group are you in?
I had Ariel and Ervik.
Okay. Ariel looks like she's disappeared from your room, but Ervik's still there.
Yeah.
You're in room 7, so I will send you there.
Okay.
All
Thank
right.
you.
Excellent. Good job. Hey, Garvit.
Hey, Professor. So we chose celiac disease.
Perfect. Excellent. What group are you in?
I was in group 9. Does it like kick you out and close Zoom when you go out of the breaker room?
No, I'll send you back to your room. So I'll send you back to your room now. So it's you and Claudia?
Yeah. Because when
Excellent.
I dropped out of the breakout room, I got my meeting got dropped.
Oh, okay. Well, you're back now though, and you got it. So I'll send you back to your room and so you guys can game plan. All right.
Okay. Thank you.
Awesome.
Hi.
Hi, Daniela.
Um,
What do you got for me?
oh no.
No.
Some
What?
of these were on our list, but we have Hashimoto's thyroiditis.
Okay. Is that your top choice?
Yeah.
All right, fun. Okay. So Hashimoto's, right?
Hmm.
Okay. Again, remember, one of the challenges is to make sure you have 8 minutes worth of material. With some of these smaller disorders, there's not as much information, so you may have to dig around a little bit more to get more detail. There should be enough there. You shouldn't have a problem, but I think that's a great topic.
Okay.
What group are you in?
I think it was 3.
So with Hannah and Samuel?
Yeah.
All right, I will send you back there. Good luck. Hey, Maya, are you there?
I just had to unmute myself. How are you?
Doing well. How about yourself? How's your husband?
Oh, it's not good.
Oh, I'm sorry to hear that.
Going to PMR tomorrow. Hopefully we're going to get an epidural injection. X-rays show slippage, a disc L2, 3, 4, 5, S1, herniated,
Wow.
bulging, stenosis of the canals. So
What was he doing?
he was doing front yard work, really. But
Oh,
then
wow.
again, we believe these are pre-existing conditions from an auto accident, DUI, that took place like 10, 15 years ago. And so he's in construction too, so that doesn't help, right?
No.
And so yeah, so front yard work and just kind of set everything out of alignment. So he's been suffering for the last 3 weeks.
Yikes, I'm sorry to hear that.
Yeah.
All righty. So what topic did you guys come up with?
So number one priority or what need is multiple sclerosis.
Awesome.
Yep.
It is yours.
Okay, sounds great.
Excellent.
Thank
What
you.
group are you in?
I was with Devlin and I think Mark or Matt.
Okay,
And Mark or
looks
Matt,
like room 4.
is it? Okay, thanks.
It's Mark.
Okay.
All right, excellent. Congratulations. All right, I'll send you back there so you guys can get to work. Hello, Ariel. Oh, you're muted.
Okay.
Excellent.
I was trying to go back to my group. It kicked me out.
Okay, what group are you in?
With Aravic and Noga, I think that's her name.
Okay, did you guys have a topic already?
Yeah, the rheumatoid arthritis.
Okay, perfect, excellent. All right, I'll send you back. So that's Aravex room 7. There you go. Hi, Amy.
Hi, Professor.
What do you got for me?
Okay, so our number one is multiple sclerosis.
Which has been taken.
Our second choice was Addison's disease.
Also taken.
Our third was celiac disease.
What was your fourth?
Rheumatoid arthritis.
And unfortunately, all of those have been taken so far.
Okay.
So again, you guys can continue to look. Let me give you some suggestions if you want.
Okay.
There is, elephantiasis or filaris. That's, where the mosquitoes inject with the larva and you get the big swelling, the big elephant feet and legs and hands and arms and stuff like that. That's always a fun and an interesting one. Hypersensitivities like allergies is a great topic for something like this. There is the severe compromised immune disorder. It's SCID. That's that bubble boy disorder. So all of those are good examples of some things. So if you want to take a quick list of what's here, those are some good suggestions, or you can find some other ones, but it looks like all of those have been taken, as well as lupus, HIV, and Hashimoto's. So there's plenty of others out there, but those are some good suggestions. And it's you and Jane, right?
Yes.
Okay, I'll send you back there so you guys can discuss some more, okay?
Okay, thank you.
All right. Hey guys.
Oh, hi.
Oh, hello.
Hey. So I'm getting down to a couple groups left. So I was curious how your search was going.
Oh, yes. I
I
think we
think
just,
we're pretty good. Yeah.
Okay, what do you got for me?
so we have Hashimoto's, lupus, Castleman's disease, and psoriasis.
So the first two are taken. I'm not familiar with Castleman's. What's Castleman's disease?
It is, go back to it. So it's a disorder that involves an overgrowth of cells in the body's lymph nodes. The most common form of the disorder affects a single lymph node, usually in the chest or abdomen.
Interesting. I'm not familiar with it. Sometimes more obscure diseases like this can be interesting. My concern is that if it's a more obscure disease, especially one with just as little of, where it's most commonly just in one lymph node, getting 8
Too hard
minutes
to
to
find.
talk about for something like that might be challenging.
For sure. We were just kind of talking about that.
Right. So whereas, what was your fourth one?
Psoriasis.
Psoriasis is excellent. That's a good skin disorder.
Very common.
There's a lot of information on there. If that's something you guys are interested in, I don't want to stop you from doing the more obscure one. If it's something you guys are jonesing to do, then I say go for it. But my concern is that you might have a hard time finding 8 minutes worth of good information. So if you're equal on those two, then psoriasis might be a better one. So do you guys want to do that?
I'm okay with that. Are you guys? I think the more information, the better.
Yeah, that's what we were looking at right now. It's like, let's go for one that's more common. So there's a lot of background information on it.
Why don't I put you down for psoriasis and you guys can continue to look. If you find something else that you really want to do that's not already on the list, or if Castleman's, if you do more research and you think it's going to be there, then switching to Castleman's will be easy. But at least if I put psoriasis down for you guys first, then that locks that in. So you guys definitely have that to fall back on. All right. So that way you guys can discuss some more. You don't have to do that. I'll keep your safety school and then you can decide what you want to do with psoriasis. All right.
Perfect. Thank you.
All right. Excellent. So just somebody come and let me know when you fully decided, but I'll put psoriasis for now.
Okay. Thank
All
you.
right. Hey, Garvett.
So after getting that done, and we like talked about, got our text and email exchange, are we good to go?
Yeah, at this point, the time is yours to use however you like. Absolutely.
Gotcha. Thank you.
You're welcome.
Yep. Have a great day.
All right, you too.
I just also want to talk about my proctorio, like the email I sent you about my test.
Yes.
Yeah, it was like the end of the exam where I really needed to use the bathroom. So I
Okay.
like,
Again, as I mentioned, the problem with that is obviously there's no way for me to monitor it while that's occurring. But so I haven't, again, like I said, my 4:30 class had the exam on Monday, so they've been my priority right now. Once I get a chance to observe the video, then we can talk about it more. I haven't had a chance to look at it yet, so I can't comment on it yet. But we'll take a look at it and we'll see. Okay.
okay, sounds good. Thank you.
Yep. You're no problem. And again, like, I appreciate you letting me know. I can't answer any of your questions about it because I obviously have to look at it and so that we can discuss what the best way to deal with it is.
Because I just didn't want to like leave it and just put it behind.
No, and I
find
appreciate
out
that. And
later.
look, I understand these things happen. It's better to try to do that, but it's also like when you're in the classroom, if you really had to go to the bathroom, then you could leave the bathroom to go to that. But normally I'd make sure that you left your phone or, things along those lines. So like I said, let me look at it and then we can discuss afterwards. Okay.
Okay. Thank you.
No problem. All right. Bye. Hey guys.
Okay. Oh, hi. Good. We're just figuring out our other options here.
Okay, well, what have you guys been discussing?
So for option number 1, we're thinking what you have suggested, elephantiasis.
Yeah.
Yeah. So
that always has some very fun visual pictures that you can use. What else is one that commonly gets done but surprisingly no one's picked is type 1 diabetes. So that's another one. Again, not type 2, but type 1 is an autoimmune disorder. So that's always a good topic. Like I said, there's that bubble boy disease, SCID. So there are some really fun ones as well. Fun not for the people who have them, obviously, but interesting topics to discuss if that's something that you guys are interested in. But yeah, if you want to do elephantiasis, then absolutely that's available.
Okay, I'm leaning more towards, I think, that one.
Okay.
What does Amy thinks? What do you think?
Oh yeah, no, I'm totally down for doing that.
Perfect. Awesome. Excellent. So obviously there are a couple different forms. So it's okay to talk about all of those, but you'll probably want to focus on the most common, which is the filaris edema one, the ones with the insect, injects the worm and all of that. So that you just don't speak too broadly because there's a couple different types. So focus on the one and really focus on that one. And there should be plenty of information for you guys.
Okay.
All right. Excellent. Well, perfect. Then you guys have your topic. I will set up the discussion boards. You guys can put your topics on there and I'll leave you to the rest of your discussion. The rest of the time is yours, however you want to use it. Okay.
Okay. Thank you.
You're welcome. All right. Excellent. Good job, guys.